COVID-19 is an infectious disease caused by the SARS-CoV-2 virus. In this video, we will explain the cell entry mechanisms of the SARS-CoV-2 virus and possible ways to block this. Here we see the virus in pink, next to a human cell in blue. The coronavirus is an enveloped, single-stranded positive RNA virus with a helical protein coat. This lipoprotein envelope has three important proteins: spike glycoproteins, membrane proteins, and envelope proteins. As we will see, especially the spike proteins are crucial for the cell entry mechanisms of the coronavirus into the human host cell. The spike protein is about 180 kilodaltons and consists of multiple subunits, named the S1 subunit, shown in pink, the S2 subunit, shown in blue, as well as the receptor binding domain, shown in orange. Before the virus approaches the cell, the spike proteins are already cleaved by the human enzyme furin. There are several domains in the spike S2 subunit that enable its binding to ACE2 receptors. The most important being the haptid repeat 1, HR1, and haptid repeat 2, HR2 residues. Upon viral entry, the viral spike protein will interact with the host ACE2 receptor through its receptor binding domain. The interaction between the ACE2 and spike RBD results in the release of the S1 domain of the spike, exposing the S2 domain and its aforementioned residues that are essential for membrane fusion. Once the virus is bound to the cell, there are two methods by which the virus can enter it. The first is direct fusion, a mechanism by which the viral membrane merges with the host membrane, releasing its content into the cell in the process. This fusion is facilitated by the protease TMPRSS2. TMPRSS2 is a serine protease that induces a conformational change in the spike protein, which tethers the virus to the host. It does so thanks to its catalytic triad, composed of aspartic acid, histidine, and serine. This combination makes the serine residues a very active nucleophile. They are located in the S1 pocket, here in red. The priming of the spike protein occurs at the S2 prime site, where the residues 685 and 686 are hydrolyzed by TMPRSS2. This step is also a possible drug target. A molecule that interacts with other residues present in the active site pocket of TMPRSS2 could inhibit this mechanism and stop the spike from being primed, resulting in a blocking of the cell entry. The second mechanism by which the virus can enter the cell is via the endosomal pathway. The virus recruits intracellular components of the host cell to construct a clathrin-coated pit in the membrane. GTPases inside of the cell bind around the pinched section of the membrane, cleaving the vesicle. This effect allows the host cell membrane to fuse back together while the resulting endosome continues into the cell. Once the endosome has formed, the release of SARS-CoV-2 variants into the cytosol is made possible by the serine protease cathepsin. The protease activates the S protein, causing a pH change in the endosome and initiating the fusion of the viral envelope with the endosomal membrane and the release of the viral material into the cell. We can see the virus membrane merging with that of the host cell. This fusion does not happen spontaneously. The S1 domain of the spike protein binds with the ACE2 receptors and detaches from the rest of the protein. The HR domain elongates into a 6-helix bundle, which is what targets the virus to fuse its membrane. After the content of the viral envelope has been released into the host cell, viral entry is complete. The viral RNA can now proceed towards the nucleus and continue its life cycle via replication, assembly, and budding.